to have you on Planetterrian Daily, episode 133 for July 27, 2026. Here's what's new in science, health, and longevity research. Virtual reality experiments show how artificial light at night changes how coral reef fish react to predators. Longitudinal proteomic profiling tracked plasma from unaffected carriers of ALS-linked genetic variants. Early protein shifts appeared before clinical symptoms emerged in the tracked individuals. Trajectory mapping allowed prediction of when symptoms would begin. The approach identifies candidate biomarkers for timing of phenoconversion. Validation in larger cohorts will determine clinical utility. The Planetterrian Spotlight adds that this work brings a temporal dimension to biomarker discovery in a disease where early intervention windows remain poorly defined. It directly addresses open questions about reliable indicators that can forecast phenoconversion rather than simply correlate with established disease. Next steps include testing whether the identified proteins can guide timing of emerging therapies in at-risk individuals. Now, shifting to a very different area of research, a new VR platform combined metabolic measurements from the University of Glasgow with an immersive system developed at the Institut de Neurosciences de la Timone and deployed at Criobe. Researchers tested how light at night alters predator responses in coral reef fish. The setup allowed controlled exposure while tracking both behavior and energy use. The work marks the first integration of these methods for this question. Follow-up studies will examine additional species and light wavelengths. On a different note, natural selection offers guidance on when suppressing fever helps or hinders recovery. The analysis links immune response patterns to historical pathogen pressures. Understanding these trade-offs can inform both individual care and broader health policies. The piece extends similar logic to crop management and social interventions. Ongoing work will test predictions in additional disease models. Next up, risk-taking in adolescence aligns with heightened sensitivity to social feedback and novelty. This pattern supports skill acquisition and independence rather than indicating simple immaturity. The perspective reframes common adult interpretations of teenage choices. Data from developmental studies show the same circuits later support adult goal pursuit. Longitudinal tracking continues to map how these priorities shift with age. Now, here's something most people get wrong about teenage risk-taking. Most people assume a teenager's willingness to retaliate against an insult or chase a risky thrill reflects an incomplete brain still learning to weigh consequences. The actual picture is different. The circuits that heighten sensitivity to social feedback and novelty are operating exactly as evolution shaped them to support rapid skill acquisition and eventual independence. Right now, while you listen, your own adult prefrontal networks are modulating those same pathways that once drove stronger exploration. One memorable feature is the speed of adolescent reward signaling, which can reach peak responsiveness before the slower regulatory systems fully balance it. The next time you watch a young person make a choice that seems short-sighted, consider that the behavior is part of a temporary window optimized for learning rather than a defect. A practical takeaway is to channel that heightened sensitivity through structured challenges such as sports, creative projects, or skill-building tasks, rather than attempting to suppress it outright. Future studies will clarify how long this window remains open and whether targeted experiences can extend its benefits into early adulthood. From evolved behavioral priorities during adolescence to engineered biological production, polyketide synthases were redesigned to generate sustainable polymer building blocks in microbes. The resulting materials allow property tuning while supporting recycling. The microbial chassis replaces petroleum-derived routes for certain plastics. Performance testing confirms structural consistency across batches. Scale-up trials will assess industrial feasibility. A small number of scarce compounds can shape community structure far beyond their concentration. The review draws examples from freshwater and terrestrial systems. These molecules influence species interactions and nutrient cycling. Identification relies on combining metabolomics with ecological surveys. Field experiments will test predicted effects of targeted removal. Researchers note that metabolomic profiling paired with field surveys offers the most direct route to spotting these low abundance drivers. Shifting focus to monitoring methods, a DNA vacuum collected genetic traces from air in 3 Danish natural areas. The method detected multiple species without direct observation. 2 studies confirmed reliable local biodiversity mapping. The technique offers a non-invasive complement to camera traps and field surveys. Expansion to new habitats is planned. From biodiversity monitoring to landscape interventions, analysis of roughly 26,400 trees showed those in treated areas were nearly 4 times more likely to survive recent wildfires. Controlled burns prevented an estimated 1,900 deaths in the studied groves. The benefit stems from reduced fuel loads that limit crown fire intensity. Untreated groves experienced higher mortality despite the tree's natural fire tolerance. Additional groves are being evaluated for treatment priority. Moving to crop genetics, researchers examined grapevines in Ethiopia's northern highlands, a region with some of Africa's oldest continuous agriculture. The vines have persisted through centuries of environmental and social pressures. Genetic data revealed distinct lineages tied to local cultivation history. The findings add to understanding of crop origins alongside teff and other regional staples. Further mapping will clarify how these populations can support future breeding. On another front, existing benchmarks for medical AI performance are described as misleading and insufficient. The commentary calls for a rigorous, task-based system to define and measure superintelligence in clinical contexts. Authors argue current tests fail to capture real-world diagnostic or therapeutic demands. Development of such a framework would allow clearer comparison across models. Work on the criteria is expected to begin with input from multiple clinical domains. Next, tissue donors often remain unaware that their cells may contribute to biocomputing systems. Current consent processes focus on traditional biomedical uses. The commentary highlights the need to address this specific downstream application. Researchers building these systems have not yet established consistent disclosure standards. Revised consent language is under discussion in several labs. Turning to imaging tools, multiple algorithms were tested against calcium imaging data collected with GCAMP8 variants. Indicator linearity emerged as a key factor in reliable spike timing reconstruction. The analysis compared performance across recording conditions and cell types. Results clarify which indicator algorithm pairs minimize timing errors. Updated guidelines for experimental design are provided. From imaging refinements to molecular separation, enantiomers that appear identical can produce different fragment counts when exposed to structured light. The method exploits handedness differences without requiring chemical labels. Experiments demonstrated separation based on fragment yield rather than traditional spectroscopy. The approach may simplify quality control in pharmaceutical synthesis. Refinements will target higher throughput. A previously considered irreversible cellular aging feature was restored in cultured human samples. The intervention used a modified enzyme targeting the specific mark. This builds on ongoing work in aging biomarkers and intervention translation. Human tissue results provide a new data point for testing which changes can be undone. Additional models will examine functional outcomes. Researchers view the cultured tissue result as one incremental test of which marks can actually be reversed. Damaged fat cells lose lipid storage capacity, become inflamed, and disappear. This process impairs whole body metabolism even without overall fat gain. The findings separate effects of excess fat from loss of healthy adipose function. Mouse models trace the sequence from cell damage to systemic glucose dysregulation. Human tissue studies will test whether similar dynamics occur in type 2 diabetes. The work focuses on whether restoring functional adipose capacity can improve metabolic outcomes independently of total fat mass. Before we go, keep an eye on results from human tissue studies testing whether loss of functional fat cell capacity contributes to glucose dysregulation in type 2 diabetes. That's Planetterrian Daily for today. If you enjoyed this, a rating or review on Apple Podcasts or Spotify really helps new listeners find the show. I'm Patrick in Vancouver. Thanks for listening, and I'll see you tomorrow. And if you'd rather watch than listen, find us on YouTube at NARA Network. Links in the show notes. This show comes to you from the NARA Network. If you want another sharp daily briefing, Tesla Shorts Time has you covered. Your daily deep dive into everything Tesla. All our shows are free at neranetwork.com. And on the website, every episode's visuals live in our free image gallery at neranetwork.com/gallery. Royalty free under Creative Commons. Download with attribution. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.